Hello everyone. Now we will discuss the different types of stem cells. This diagram will eventually show the entire range of development from fertilized egg to mature cell types in the body. Each cell in 8 cell embryo, here in red actually, can generate every cell in the embryo as well as the placenta and extra embryonic tissues. These cells are called totipotent stem cells. Why are they called totipotent? Because one red cell can potentially make all necessary tissues for development. During in vitro fertilization, can parents choose whether their baby is going to be a boy or a girl? Yes. There is a widely practiced procedure called pre-implantation genetic diagnosis, where one cell is removed from the 8 cell embryo and its DNA is examined. What might you look for when trying to identifying the embryo sex? If there is an X and Y chromosome, it's a boy, and if there are two X, it's a girl. The parents can decide whether to implant it. Also, parent with a genetic disease might want to see if their baby has an identifiable genetic disorders and decide whether to implant based on this information. Pre-implantation genetic diagnosis does not destroy the embryo. Scientists are attempting to adapt this implantation genetic diagnosis procedure and use it to create a stem cell line from one single totipotent cell without destroying the embryo. The embryonic stem cells inside the blastocyst, here in purple, can generate every cell in body except placenta and extra embryonic tissues. These are called pluripotent stem cells. Why? Because they can differentiate into all the 200 plus cell types in the body, but they do not form the placenta. Pluripotent stem cells can be isolated and grown in culture or left to develop into more specialized cells inside the body. Adult stem cells or tissue-specific stem cells have restricted lineages. Adult stem cells show up when the 3 distinct layers form in the 14-day-old embryo and are present in the fetus, baby, child, and so forth. Adult just means they have gone further down their lineages pathways than the initial stem cells in the embryo. They are called multipotent stem cells. Multipotent stem cells because they will only become mature cells from the tissue in which they reside. Adult stem cells are present throughout your life and replace fully matured, yet damaged and dying cells. And totipotent stem cells come from embryos that are less than 3 days old. These cells can make the total human being because they can form the placenta and all other tissues. Pluripotent stem cells come from embryos that are 5 to 14 days old. Embryos and fetuses that are older than 14 days do not contain pluripotent cells. These cells can form every cell type in the body but not the placenta. Multipotent stem cells are also called adult stem cells, and these appear in the 14-day-old embryo and beyond. At this point, these stem cells will continue down certain lineages and cannot naturally turn back into pluripotent cells or switch lineages. Adult stem cells are found all over your body. Here are a few examples of places in the body with stem cells. Who here has been told that brain cells never regenerate? Whoever told you that was misinformed. Relatively recently, scientists discovered that in 2 specific parts of your brain, neural stem cells divide and differentiate to become neurons and glial cells, which support the growth of neurons. Without neural stem cells in the hippocampus, you would probably not be able to learn or remember. The top right picture is a cross section of the rat hippocampus, and neural stem cells are the blue dots which divide and differentiate to form mature neurons, in green particularly, and astrocytes in red. The bottom right picture is of cultured neural stem cells, just plain blue dots, and derived from those stem cells, neurons, blue dots surrounded by red. and oligodendrocytes, blue dots surrounded by green. In skeletal muscle, stem cells are activated immediately after an injury to help replace damaged muscle. Imagine the muscle fibers running down Arnold's bicep are like a bundle of spaghetti. We are going to take a cross section of this muscle. So break the bundle in half. Break the imaginary bundle in half, and we are going to look at the tips of the muscle fibers. Turn your hand so that the end imaginary spaghetti bundle is facing. Who can tell me where the stem cells are? The big red circles are healthy muscle fibers, the green cells are newly regenerated muscle fibers, and the teeny pink dots are muscle stem cells, also called satellite cells, because they hang around the sides of muscle fibers. The teeny pink dots eventually turn into the green cells, which add together to form the red muscle fibers. When Arnold lifts weights, his muscle stem cells proliferate and differentiate into new muscle fibers, which add on to existing fibers to increase the size of his muscles. This is part of the reason why muscles get bigger with exercise. Another part is that the muscle fibers themselves actually grow bigger. Cells stick and respond to molecules embedded in their extracellular environment. They also respond to chemicals or molecules floating around in the liquid surrounding them. Cells can feel and communicate with each other and also can respond to forces. How might an embryonic stem cell respond if it touches a bunch of muscles? It might differentiate into a muscle fiber. How do you think that same stem cell would respond to culture with a bunch of neurons? It will turn into a neuron. Here, this stem cell is going to make a decision to self-renew or differentiate based on the individual components and combinations of these factors in the extracellular environment. So, how do stem cells know what to become? All cells in a person share the same genetic code or genotype, given that their DNA sequences are the same. Why are your eye cells differentiate from your nose cells? If you cut your hand, how do the cells know how to make more hand cells, not eye cells? Well, that environment outside the cells, also called the microenvironment, influences the switching on and off of different genes. Stem cells live in a special microenvironment called the stem cell niche. Niches consist of a multitude of factors that can influence cell behavior. The microenvironment induces changes in gene expression that alter the physical characteristics or phenotypes of the cells. This should sound familiar to everyone since this is the central dogma of biology. That is, gene expression, DNA to RNA to protein, drives the presence and levels of certain proteins which dictate the cell type. Finally, cell types constantly contribute to the components of the niche. For example, fibroblasts secrete proteins that contribute to the extracellular matrix and your connective tissues. Induced pluripotent stem cells, that is IPS cells, are another type of stem cell that is useful for studying disease in vitro and may yield cells that can be used for tissue therapy. One way to create these cells is by genetic engineering to insert DNA sequences that can eventually alter the cell type. Another is to give them signals from their environment, pharmaceutical molecules that influence gene expression. Creating induced pluripotent stem cells is like turning back the clock in a subset of cells. This method creates pluripotency rather than directly harvesting or cloning embryonic stem cells. In induced pluripotent stem cell technology, you first isolate and culture skin cells from a patient. Secondly, you introduce 3 or 4 pluripotency genes into the skin cells by using an engineered virus carrier. The expression of these genes regenerates the stem cell phenotype. The viruses simply deliver the genes of interest and are themselves engineered not to be harmful. Here the red cells indicate the cells actively expressing the 4 essential stem cell or pluripotency genes. How does this transfer of pluripotency genes from virus to skin cell actually happen? And how do these genes start producing proteins that physically alter the cell's characteristics? What exactly is a pluripotency gene. Let's pause here and talk about this, then we will return to the rest of the procedure. A bioengineer would start out with the DNA transcripts of 4 genes known to promote pluripotency. These genes code for proteins that regulate the expression of other genes. These special proteins are called transcription factors or master regulatory proteins that bind to and regulate the transcription of genes that say code for the ability of a cell to proliferate. So something called a pluripotency gene is actually DNA that codes for proteins that regulate the production of other proteins which give the cell characteristics of pluripotency. If you want, the bioengineer takes those 4 genes and packages them into empty viruses. These viruses are not the infectious kind. They simply deliver their DNA contents to the skin cells and cause their genes to be incorporated into the host genome. Those transcription factor genes are now expressed by the cells, changing the phenotype and thus reprogramming these cells into embryonic stem-like cells. So back to the procedure. In the third image, after the skin cells have been genetically engineered, you harvest and culture them according to the method for embryonic stem cell culture, that is together with feeder cells. Finally, through this process, a subset of cells generates embryonic stem cell-like colonies called induced pluripotent stem cells. One of the scientists who pioneered this technology is Dr. Shinya Yamanaka in 2006. Already scientists at Harvard and other institutions have used this process to create over 20 stem cell lines that model different genetic diseases. They do this by starting with the skin cells or other fully mature cells from patients with the diseases. What are the pros and cons to iPS cell technologies? Pros are that the induced pluripotent stem cells would be genetically identical to the person who donated the skin cells. So any cells derived from these iPS cells would not be rejected by the patient's immune system. Ultimately, this would be a move toward personal cell-based therapies. Another advantage is that you do not need to use an embryo to get pluripotent. So what about the demerits? Any cell created from a patient with a genetic disease as opposed to an environmentally caused disease would carry the same defective genes. So if a Parkinson's patient needed new motor neurons, iPS would not work because the stem cells would display the same symptoms. Another disadvantage is that one of the pluripotency genes is also a cancer gene. This gene is used to stimulate the cells to start dividing again, and it must be controlled. If some of the iPS cells turn into cancer cells, then patients might develop tumors as a result of the transplant. Also, since viruses insert these pluripotency genes randomly into the genome, you might be interrupting the normal sequence of an important gene and cause an undesirable mutation. Scientists have already found substitutes for this cancer gene as well as alternative methods of delivering and expressing these genes, which could eventually make this process safer for transplantation. For now, these cells are perfect for making and studying diseases in a dish to promote basic research. But many important details need to be worked out before this tool is used as an actual technology in the clinic to treat patients. So, if growth factors and hormones affect stem cell functions, then our lifestyles, our experience, and behaviors are also likely to influence homeostatic stem cell related processes. For example, regular cell turnover in blood and skin, wound healing throughout the body, and even our sensory and cognitive abilities when it comes to adult stem cells in the brain. So not surprisingly, what is good for your body is good for your stem cells, and so what is good for your stem cell is also good for your body. Why? Because the regulation of stem cells not only plays a really important role in various disease states, but also these proliferative cells actively participate in maintaining our overall health. If you are good to your stem cells, they will be good to you.